So, Helen, I left you
So how do you share
one
this
page.
link?
Mary, you can take that one. That's okay. There's one for everybody. It's just, we're not going to
It's
use
live
it today.
now.
I want to talk about it though before we start.
Oh, okay. You share it and let me know if it's working.
That's Stephen.
Stephen and someone are both at St. Chris.
So they're supposed to be back here though, right? They am tomorrow, but this is a mandatory lecture. Everybody except people out of town need to be here.
Yeah, would we be able to get an email there from you, like making sure that because they don't let us go, like
You come if that's what you need, yes. But that's the people from St. Chris and the people from Jefferson have to return here for this lecture for basic neuroscience. This is a cannot miss lecture.
I will stress on that. Last time Annie was trying to come back from there, she waited for the bus and missed it because she doesn't have conveyance. So
you have to leave early, and I mean, but this is mandatory.
Like Maya said that she wasn't going to move the patient in a status.
Okay, where's it like?
It's just for the neurology residents, actually. The others can look at it. There's nothing there. It's just to introduce a topic of discussion. So, like you said, is with the patient in status.
Maria.
Maria. And so, Eli, and where's someone?
Someone in Stephen or both at St. Chris.
Sorry, where?
St. Chris.
So,
I'll let them know to come for tomorrow. We also have a lecture.
Yeah. And yeah, where's Eli? No? And who else? Pavana's on nights. Maria,
Sorry,
that's okay.
Maria's the patient that is in status right now. She's just added on.
She could, and that's fine. But I mean, she could just ask who's the consult attending.
Dr. Bradley.
I think he can manage status.
Yeah, it was in the AD with me.
So ideally, the attendants are supposed to take over the pager during noon.
Yeah.
But I think Dr. Prabhu's
he was done in the AD same 3 consults with me.
Say again.
He was in the EDC consults with me also. And I'll tell Maria to,
Yeah,
I'll tell Dr. Bradley to see if he
right. You can relieve her.
can. Absolutely.
He could manage that. So have a good time.
Yeah,
He's
I'm
an
sure.
epileptologist. It's all right. He can do it.
And the EEG, I think Maria was pushing the adamant. She said that the EEG is down a lot.
It will be good for Dr. Bradley to interact with the ED.
She said that she's in the book. She's in the patient.
Dr. Newman, are you gonna have the movement disorder clinic after the January?
Say that again. Yeah.
Movement clinic.
Yeah.
Is it gonna be part of the clinic?
Yes, there will be a
Schedule.
movement disorder clinic. I don't know the schedule. So the schedule doesn't include movement disorder, but when Dr. Ndavia is not available for neuromuscular, then we're going to have movement disorder. That's where it is. If Dr. Ndavia is either away or on consults or that sort of thing, then we'll put in movement disorder. So for you want to just pass one of those around to everyone and give one to Eli. Okay. So I think that, I've been thinking about, so first of all, the comments from last Friday's residency committee meeting and pertinent to this course, the basic neuroscience course. And the analogy of it being a language struck me very strongly. The thing is, so there is an internal logic to each of these courses. And the internal logic, it's something that you don't make explicit, because you're never really dealing with the logic directly. You're always dealing with a specific example of the logic. And each of the lectures, though, shares a framework, if you will, a logic is all that I can say. That is, there's a language of neuroanatomy that is separate from the information of neuroanatomy. It's a way of thinking, okay? And if you disperse the disciplines and do them around topic like movement, the basal ganglia movement or something like that, there's a certain logic to doing it that way. That's called the systems approach, okay? And there's a logic to doing it that way. But what it doesn't do is it cannot convey to you the internal logic of the discipline of science. Each of these different science disciplines has their own way of thinking and speaking, okay? And if you focus on the system, you never see that. The rest of your life, you're going to look at it as a systems, okay? So you have the rest of your career to look at it from that point of view. But you will never, not ever again in your life, have the opportunity to learn the language of the science discipline that creates that information. Now, that's still abstract. So look at, when I read this article, this is from this week's Neurology or last week's Neurology. And this is an article by Eduardo Benarroch. And if you don't know Eduardo Benarroch, you really should look him up. He writes this series of articles on basic neuroscience for neurology. They are stunning. I've written to him and told him he should just collect them and make a book out of them. They're fantastic articles. I want you to read the first two paragraphs. I mean, I literally want you to take 2 or 3 minutes here and read the first two paragraphs. And just, can I have a copy also, please? Yeah, there should be enough copies for all of us. Thank you. That's what I'm saying. This is Eduardo Benarroch. He's from Mayo Clinic, is where he is. And I don't know what his field is. I didn't know him as a clinician. I just know this series of articles they're all fantastic. Every one of them is fantastic. Okay. So without I don't. So what's going on here? Okay. So first of all, my point in putting this in front of you is that without this course, you wouldn't stand a chance in the world of understanding a word of this. The language, the vocabulary that he's using here is directly out of this course. Okay, this is new. We did not cover this topic in this course because this is new information. But this information is being put out here by Eduardo because it is now believed to underlie the degeneration of ALS and frontotemporal dementias. Now, I have said many times that this course is not, there's no clinical aspect to this course. And that is true, but not quite true, because the idea of the course is that it will give you the foundation that you need to read pathophysiology, really, for the rest of your life. Okay? So this is a time to learn things that you're not going to learn on your own. You're not going to stop and think about the mechanism of G proteins or the mechanism of cytoskeletal transport and the individual constituents of and the concepts that are involved in all of these things. And you're unlikely to look at synapse in the detail that we did. While there's nothing clinical, today's lecture, first of all, is going to be quite clinical. But while there's nothing clinical in the individual lecture, they lay the foundation for understanding new information. And if you don't have that basis, then you don't have the basis for understanding the information that's going to be emerging over the next 40 years of your career. And that's all there is to it, okay? So I think that you want to learn. I think that it is my belief that if you want to not just be a modern neurologist today, but you still want to be a modern neurologist 30 years from now, you're going to have to understand the basic neuroscience of the nervous system. And if you don't understand that, then you will not be prepared to understand and utilize the concepts that emerge over the next 3 or 4 decades. And that's all I can tell you. And I think it's important that you learn it. To illustrate it, what I would like to propose that we take on this article as a mini symposium. I propose that we take 2 hours sometime, I don't know when, more than a month from now, maybe in the new year, and that we have a mini symposium on this article. And I would like to divide up topics. Each resident will be given a roughly 10 to 15 minute presentation reviewing the basic neuroscience so that at the end we can assemble this into a rational understanding of this article and go all the way through the article and present the article piece by piece. And someone will talk about chaperone proteins, someone will talk about stress proteins, okay? Heat shock proteins, that sort of thing. What are we talking about when we say a heat shock protein? That is not something that we have introduced in this course and won't. What about folded proteins? How are folded proteins handled by the brain? Okay? And then assemble it, okay, into a discussion of this article. Because this article is an important article. This article, okay, is saying that this receptor, the sigma R1, the sigma 1 receptor, basically appears to underlie some of the most important diseases that we face that are not understood and for which we have no treatment at the present time, and that they suggest treatment avenues for future research. That's pretty damn good. And I'll say again, I think if you didn't have this course, you would not be able to read this article in any meaningful way. Okay? So that's where I am. That's where my head is on this topic. And it's why these lectures are mandatory as well. And when that includes people at St. Chris and at Jefferson. Okay. All right. Amino acids. So this is what we're talking about: nitrogen, ammonia, and amino acids. And it builds on last week's lecture. And by the way, Gary Horowitz thought that we had it backwards, that since today is Halloween, and in honor of the bag that's on this table, today should have been a discussion of glucose metabolism. It's not. All right, so we're going to talk, this is our outline, we're going to talk about I'm going to remove this or hide this. So we're going to talk about nitrogen cycle in the atmosphere, actually, in the world, and ammonia as a molecule. We're then going to look at the amino acids in the brain and the liver, transaminase reactions. We are not going to consider the urea cycle in any manner that you would use for inborn errors of metabolism. We're going to set it in context, and that's it. We're then going to look at the role of glutamate in the brain, and you had a hint of this last week. And then we'll look also at the entity of hepatic encephalopathy. All right, the nitrogen cycle. Again, hello, Maria. Good, we're just getting started. So the nitrogen, so right, 78% of the atmosphere is nitrogen, okay? You're not really aware of it. We don't feel different if we, because of nitrogen, if we hyperventilate, we don't, nothing. Nitrogen in the form of N2 is not really, the only thing it does is limit the amount of oxygen. Other than that, it has no other interaction with us. So we have to get it into another form in order for it to be biologic. And the biologic form is generally ammonia. All right. So you get ammonia in the world nitrogen cycle through soil organisms primarily. You'll see that there's lightning can also generate ammonia, but it's not a reliable thing. So we're going to be talking about biologic nitrogen, ammonia, which is through soil organisms. The process by which soil organisms take molecular nitrogen and convert it to ammonia is called nitrogen fixation. This is carried out by bacterial nitrogenases, is what they're called. And they also can be used to form amino acids directly. By the way, you can take a stroke of lightning if you have water, carbon dioxide, and nitrogen in the atmosphere, and you hit it with an electric discharge. What you get is tar at the bottom of the flask, and that tar is peptides. All you have to do is hit it with lightning. That was C.N. Matthews discovered that in about 1967, approximately 1968. That's literally what he did. He was trying to create the reducing atmosphere of Earth before there were plants, and he just discharged the thing, and lo and behold, he didn't get individual amino acids, he got polypeptides. That's what, all kinds of peptides, all including most of the amino acids were generated. It freaked everybody out when that happened. Anyway, nitrogen enters the human body either as free amino acids or as protein in our diet, or through ammonia generated by intestinal tract bacteria by cleaving amino acids or polypeptides. And this is a reminder that the gut is, strictly speaking, from a topology point of view, outside of our body. We carry that gut around in it, but from this end to this end is not connected to the rest of the body. It's a tube that runs through our body. It's outside our body. There are more bacteria, there are more cells in our gut than there are in the rest of the body put together. They have their own biome, okay? And I frequently say that you are what you eat, because these guys can make all kinds of peptides, including peptides that are active. And so if I keep thinking about almond joy in there, my biome keeps saying coconut. I would like some coconut. Okay. This is your atmospheric nitrogen. We're just going through this. The main source of nitrogen, again, is decomposition of plant and feces from animals by a variety of decomposing single cell organisms that eventually generate ammonia, which is what we're going to use. The rest of it is less important to us. While you can generate ammonium and ammonium nitrate via lightning strikes, that's not gonna, you can't rely on that. It's only there during the wet season at certain times of year. You can't, that's not adequate. The biology requires a continuous source, which is, I need to hide this, which is nitrogen fixation by the nitrogenase reaction and through the soil organisms. Now, on this slide, there are a couple of things. First of all, the lightning strike. So think about the energy that's involved. So ammonium nitrate, in case you don't know, is what you make explosives out of. Okay, so if you're a terrorist bomber, this is what you load your truck with, is ammonium nitrate. And that tells you that the energy contained in this molecule is huge. These bonds are high energy bonds. And if you break them, you have a lot of energy. Or if you want to make them, you're going to need to consume a lot of energy. So look at the nitrogenase reaction, okay? 8 protons, 8 electrons, a nitrogen molecule, and you end up with 2 ammonia. Look at the consumption. You have to spend 24 ATPs to make this little molecule. That just tells you how much energy is required. I mean, we've been through this in the body, right? To get over the barriers, we've spent an ATP, okay, here or there. We threw an ATP in and got over a barrier. 24 ATPs here, okay? This is not something that happens spontaneously, by the way. So you're gonna, over and over and over again, I keep trying to get you to think about evolution and the origin of everything. In this case, we're talking about the origin of life, okay? How did it get to this point, okay? It's an interesting thing to think about. Now, when you look at ammonia, ammonia is a very interesting molecule. I mean, I just think from a chemist's point of view, PChem point of view, this is a really interesting molecule. It has this, so it sits there as a trivalent, and the nitrogen, so there would be 8 electrons, right, to complete this valence shell. There are 2 shared with each of the hydrogens, and then there are 2 unshared electrons to this. So you have this electron cloud. That's sitting out there ready to go and grab a free proton, okay? And you end up with ammonium, all right? And that's a tetrahedron. That's a pretty little molecule right there. There's your tetrahedron. It's a perfect tetrahedron. There's a lot of symmetry to it. It's just a cool molecule, okay? It has a pKa of 9.24, which means at physiologic pH, this is a strong base. This is a very strong base. And at pH 7.4, all of it's going to be protonated. That is, you're not going to run into very much free ammonia. You're going to have ammonium, and ammonium chloride is going to be the salt that you run across everywhere. In its hydrated state, it's roughly the same size as water. Okay, that's important in terms of how it gets across membranes. So it's roughly the same size as water. Its dielectric constant of 16.5 compares very favorably to ethanol. So if you have a sense of miscibility, if you will, of alcohol, of ethanol, in water, in oil, it's not a bad in between. And so is the ammonia. Ammonium will not have that dielectric constant. Ammonium is going to be a salt, okay? So again, so that means that this thing is flitting around in different forms, all of which are interesting and biologically active. You have this equilibrium that goes on all the time. And this is just a simple diagram to show you at pH 7.4, some of your water is dissociated, right? It dissociates at 7.0 into proton and hydroxyl ion. At 7.4, there's a fair that, which is more basic. So some more of the water dissociates into an acid state. So there's a lot of it. It doesn't take a lot for this energetic pair of electrons, which is hydrated by water, right, to just clip off a proton and say, you come over here, okay? You stabilize me, okay? And then you're left with a hydroxyl ion, which is why this is a base, okay? Now, let's talk about how ammonia or ammonium gets across the blood-brain barrier. We're going to come and go on the brain. Ammonia can cross the blood-brain barrier either as ammonium through potassium-like channels. So look at the size of the unhydrated ion. Potassium is 1.38 angstroms, and ammonium is 1.48. So these are fairly similar guys. Sodium might be 1.12, very different. Calcium even smaller, 0.96, something like that, because the charge makes it very condensed, okay? Potassium is not, because it's up 19, right? So it's a loose electron shell, it spreads itself out, the nucleus doesn't hold it very tight, and so ammonium can actually travel through potassium channels. It's a salt, right? The ammonia can go through gas channels. There are gas channels for CO2, all right? So ammonia, all right, with its dielectric constant of 16 can, insinuate itself right to the edge of the plasma membrane and then cross via this class of channels that are designed mainly for CO2 transport, and the ammonia can join it. And the third way that ammonia gets into the brain is glutamine. Why do we need ammonia in the brain? Because the brain, two things. The brain, has to make its own amino acids. All right? The brain does not use dietary amino acids. They don't cross the blood-brain barrier. So the brain is, and if it wants to synthesize stuff, it's going to, and also neurotransmitters, especially glutamate and GABA, are both really derived from glutamate aspartate. They're all serine. I mean, all these guys are amino acid neurotransmitters. So the brain has to be able to regulate it. So it doesn't want to deal with the rest of the bodies. It wants to have its own. And the question is, how does it create all of these amino acids? It's going to make them itself, except of course for the essential amino acids. But in order to make amino acids, it has to engage in nitrogen metabolism. It has to be able to import nitrogen. It has to be able to excrete nitrogen when it has more than it needs. That's what this lecture is focused on. Okay? So this is how it gets in. Ammonium through potassium, ammonia through CO2 gas channels. And then the other one is glutamine. Glutamine is the neutral amino acid transporter. You're going to see this over and over and over again in this lecture. And glutamate, as you will recall from last week, I hope, is the center of brain metabolism. It's not just a neurotransmitter. It sits at the interface between all of the oxidative pathways and all of the amino acid neurotransmitter pathways, because it and glutamine are how you move nitrogen in the brain. Okay? They are how nitrogen is dealt with. Yeah, the fastest, which would be the fastest?
I'm just thinking your synthetase.
This is a high, so that's a good question. Transport has two different things. It is both the volume or the capacitance as well as the avidity with which you transport. Those are different things. So you could have, for instance, I always say when I used to have to create, as part of my research, I had to create a lyophilized phosphatidylcholine. And you had to create a high, really intense vacuum to do it. So you had this jar filled with liquid. You couldn't use a high intensity pump. A high intensity pump would gag on half a liter of solvent. So what you did, you started with a low affinity, low pressure, but high capacity pump. You just watched the stuff boil off. And then in about a minute or two, you would hear it switch to a different pump. And now you have a vacuum, okay? And that vacuum was a much more high intensity. There are several times that you use, for instance, we saw calcium metabolism by the mitochondria. The initial, after calcium is released into the synaptic bouton, the mitochondria has to take it back up again. It has to go from 10 to the minus third to 10 to the minus seventh, okay? Like I said, if it tried to use a 10 to the minus seventh capable pump to suck these 4 orders of magnitude out of the cytoplasm, it would gag on it. It couldn't do it. So first you have a high capacity, you bring all the calcium back in and you take it down to about 10 to the minus fourth. And then that last order of magnitude, you use a different pump with a very high affinity, but a low capacity for dealing with the calcium. So the main way that you move large volumes of ammonia are through these potassium-like channels. This is a much smaller pathway because there's not a lot of free ammonia at pH 7.4 with a pKa of 9.24, right? Okay. This diagram just shows you, and it also shows you an important feature, and that is when we're doing all of this stuff that we're doing, whether we're using glucose, whether we're making amino acids for proteins, or whether we're using more neurotransmitters, we are in and out of the mitochondria all the time, okay? The mitochondrial pathways, even in the bacteria, these nitrogenase pathways are shared between the cytoplasm and the mitochondria. And if you remember that I think of the mitochondria as a symbiote that's living inside our cell, then that means that this process, whereas we saw glucose, You saw, well, you did see it with glucose. We saw glycolysis in the cytoplasm and oxidative phosphorylation in the mitochondria. There's something analogous to that here, okay, in that a lot of the handling of this will occur in the cytoplasm, but a lot of it goes on in the mitochondria. So the mitochondria is essential to the amino acid metabolism. Remember, glucose and ammonia are, these two together make a section of this course. This is what I call intermediary metabolism. They go together. And they're the only two that belong together, in my opinion. Right, let's get the nitrogen now into protein. So glutamate dehydrogenase and glutamine synthetase, all right, are found in all organisms, all biologic organisms, and affect the conversion of ammonia into amino acids, okay, and to glutamate and glutamine, respectively, okay? So these two enzymes. The amino and amide groups from these two substances freely transfer to one another between the carbon skeletons and the amino acids. And that is done via the glutamate-glutamine couple. Right, let's just look at a few structures real quick. This is an amino acid. There's an amino group, and here's a carboxyl group. Okay, this R, and there's always a CH in between them, and then R is anything that gives the amino acid its name. They are ionized at pH 7.4 because this character over here is not going to exist. It's just like the ammonia molecule. It's going to protonate itself, and this guy also isn't going to exist. It's going to release its proton, and so this is usually charged. All right, this is an amide linkage. Here's the amide linkage. Nitrogen, so carbon and carbon on either side of it, and then you have a carboxyl group and a nitrogen group. This is amide. This is how you make polypeptides. They are amide linkages. And this is a good example of the reaction where you just pull off a piece of water, okay, and you link these two guys together. And you just throw out water. Nice and simple. Glutamate dehydrogenase, okay, goes in, it's a reversible enzyme. And it goes between glutamate and alpha-ketoglutarate. And alpha-ketoglutarate from the tricarboxylic acid cycle, okay. We talked about it a lot last week, and we're going to talk about it a lot now. And you also see that when you run it in this direction from glutamate, which could be dietary, you could have a lot of excess glutamate, and you take it to alpha-ketoglutarate for use in a carbon skeleton. You could go ahead and use glutamate for energy. You don't have to use it for amino acid synthesis. You could just use the glutamate for energy. And it's beautiful for energy because look what happened. When you use the glutamate dehydrogenase in this direction to make alpha-ketoglutarate, which you're going to then consume, you make NADH. So there's 3 ATPs right there. Okay, but you've just generated an ammonia. And if you're doing this in the brain, you now have ammonia floating around the brain, and you're going to have to get rid of it. Okay, so that's going to be a problem. All right, so the forward direction converts ammonia and alpha-ketoglutarate to glutamine, and the reverse direction takes it, and just what we just said. This is a great way of going when you need energy. You can use your proteins. That's called catabolism. All right, glutamine synthetase, is, so glutamine is the major storage form. So that's how, all right, let's get that idea straight. When you don't have a lot of free ammonia or ammonium in the brain, but you want to have a reserve for amino acid synthesis or glucose utilization or whatever you're going to do. So you want to have a reserve. The reserve storage form of ammonia in the brain is glutamine, which will be stored in astrocytes. Okay, we'll look at that. We'll look at cells in a couple minutes. The nice thing about glutamine is it's not ionized. It's a neutral compound. And it is relatively non-toxic, even in fairly high concentrations. You can have a lot of glutamine. And as you'll see in a minute, it doesn't contribute a lot of osmoles either. So this is a pretty good storage form. We'll come back to that. The enzyme glutamate, so we've talked about glutamate dehydrogenase and glutamine synthetase. This is a third enzyme, separate. So glutamine synthetase is not reversible. It had an intermediary with a phosphate group on it. You're not going to be able to go backwards through that. Okay, this is a good idea, a good example of the language that I'm trying. So I have been talking about energy flow, and I haven't really talked about energy flow as a concept. maybe a little bit, but not a lot. But in every time I keep showing you specific stuff, I keep trying to emphasize it. Just talked about it took 24 ATPs to make the ammonia molecule. I'm trying to give you a sense of energy flow in the body. That is something that you won't get unless we bring these lectures together around the chemistry. Okay, this concept of energy flow never appears otherwise. Okay, so this is just part of the logic of energy flow. You don't have to remember it. It's just if you have an intermediary, you're not going to reverse it. Okay, it doesn't behave that way. Flowing. Okay, so it needs a separate enzyme to go backwards. This is glutaminase, which will then convert glutamine to glutamate and the nitrogen, and there you go. All right. And this is nice because it shows you the nitrogen and the one that is cleaved off. Here's your amino acid group. So that's in violet. Nobody's playing with it. Starting with an amino acid and you're ending with an amino acid. Nobody's fooling with the amino acid. That's a constant. It's the head group was where all the action is occurring. All right. We're not really to the brain yet, except we talked about blood-brain barrier. But let's look at the rest of the body for a minute. Amino acid absorption occurs primarily in the duodenum, and there are lysates of proteins made by the pancreatic enzymes and the gut flora. All right, they are generally actively transported out of the gut and through the brush border, and they go into the portal circulation and onto the liver. The flora plays an important part. I keep saying that they are. We understand it. They are communicating with us. They move us to eat. They tell us it's hungry time. They tell us they want more protein in their diet. There's lots of stuff going on between our gut who live outside of our body. All right. The liver then contains the glutamine synthetase and the glutaminase. So it has both enzymes to run between glutamate and glutamine. Now, it also has all the enzymes that it needs, and it's the only place in the body that does. It's the only organ that does. It has all of the enzymes for the urea cycle. And we're not going to get into the urea cycle in this lecture, but the thing is that the urea cycle will consume bicarbonate. Right? It consumes bicarb. And therefore, at times of acidosis, when the body wants to conserve bicarb, the liver actually switches to glutamine metabolism as its way of excreting ammonia instead of making BUN. See, so BUN actually goes down. The ammonium will go up a little bit, but if you measured the glutamine, it would go way up. Well, you go, wait a minute, and now I got all this glutamine floating around, but I haven't gotten rid of the nitrogen. And of course, the answer is no, but the kidney has tons of glutaminase. So as soon as the glutamine hits the kidney, it's cleaved. The ammonium is already in the urine. So it's out in the filtrate, right? And it pulls the glutamate back because it doesn't want to lose that alpha-ketoglutarate skeleton, okay? And it, pisses out the ammonium, okay? That's it, it's done. So the urea cycle is the major way of getting rid of nitrogen, okay? Excess nitrogen. There's no question, it's the major way, but it's not the only way. And when you approach your patients who are acidotic after cardiac arrest or in sepsis, you need to start thinking about the derangements that are occurring. And I don't know what you think about when you think about acidosis. When you see a pH of 7.1, it's like, what does that mean to you? What is that, what are the consequences of having a pH of 7.1? Okay, every organ is influenced by this. Its function isn't just impaired by the acidosis. I think that's sort of the first thought as well. It won't run as well as it normally does because its pH is off center here. But it's way more than that, because the organ is going to respond to the acidosis. It's not just going to not function well. It's going to change its function because it's trying to deal with acidosis as a body. Okay, and one of the ways it will do that is the liver will simply stop creating urea and switch to a different form of metabolism. Now, you got to go, whoa. Which now, if you start to couple renal failure to hepatic failure, okay, then we're really going to have trouble. Okay, so there's a major interaction that goes on between those, ending in encephalopathy, of course. The essential amino acids, the only thing I would say is there are 10 essential amino acids. We cannot make them in the body. If we can't make them, the brain can't make them either. So they come in through dietary, and that's that. These are the branch chain amino acids, your aromatic amino acids, and sulfate-containing amino acids. The body cannot make any of those. It can't make branch chain, it can't make aromatic, and it can't make sulfate. And so all of those are going to have to come in through our diet. And these are your essential amino acids because they have one of the properties that I just said. Okay, and everything else is easily made basically through glutamine. All right, now, I put these terms in to try to help you guys, but try to introduce a little clinical here so we keep you guys awake. But the removing nitrogen, okay, how do we get nitrogen off? So if we have excess protein, I just had a hot dog, right? I did, I had a hot dog. I have a lot of, I switched, for whatever reason, I switched to a lot of protein in my diet. I go out for steak, seafood tonight, a lot of protein, right? So now I'm going to have an excess amount of protein, and therefore I'm going to have an excess amount of nitrogen because I can consume the protein for either fat storage, unfortunately, or for just energy consumption, right? Okay, but I'm going to end up, so how do I get the ammonia off the amino acid so that I can use the carbon skeleton of the amino acid for energy consumption? So before I excrete the ammonia, that nitrogen, before I excrete it, I have to get it into a form that is excretable, okay? Which means I have to get the nitrogen the heck off the amino acid. That is done by transaminase reactions. There's no cleavage. Cleaving it, why not? Because it would be a horrible waste of energy. We just spent 24 ATPs per bond, per molecule, therefore 8 ATPs per bond. Why would you just cleave this thing and burn it? Okay, generating heat. Okay, you don't want the heat, and you do want the energy. Therefore, what you want to do is get rid of it in a nice energy conserving form. So you transaminase to glutamate. So look at this. Here's glutamate. Here's oxaloacetate, right? This is AST, okay? This is serum GOT, the glutamate oxalate transaminase, okay? That's what we call it. But in the brain, of course, it's not. And you go from glutamate becomes alpha-ketoglutarate, and oxaloacetate becomes aspartate, but I could also run this in the opposite direction if I wanted to. All right? This is a transaminase reaction. Hardly any energy is expended in either direction. I'm just moving a nitrogen from one amino acid to another amino acid. What I'm really doing is taking a 5-carbon skeleton and a 4-carbon skeleton and switching who has the nitrogen between them. That's really actually all that I'm doing in the SGOT reaction. And you can do the same thing with SGPT, which is a glutamate or glucose pyruvate transaminase. It's a glutamate, and in this case, it's really an alanine. That's why it's renamed. ALT is actually a better name. It's an alanine lactate transaminase is what it is. And glutamate and alpha-ketoglutarate are the intermediates. So you're doing the same thing. Here you are in muscle, you generate lactate, you get rid of the lactate, not as lactate, actually, but as alanine. You throw the alanine into the blood, the liver picks up the alanine, and it can convert it all the way back up to glucose if it wants. And so you see you have these cycles between the producers and the consumers, okay, that are constantly going on, and they're all designed to conserve the energy, okay, as well as, in this case, to move the nitrogen is the purpose, but to move it in such a way that you don't waste energy. Here's urea, a very intriguing molecule in its own right, beautifully symmetric, right? About 80% of the excreted nitrogen of our body is produced as urea, which is exclusively out of the liver, okay, split between the mitochondria and the cytosol. And basically what you have is the urea cycle, where arginine comes in from the diet or other protein breakdown, and it's cleaved by the enzyme arginase to create urea and ornithine. And then once you have the ornithine, you remake the arginine, okay, and that's the urea cycle. Excuse me.
Sorry, sir.
You beginning and end with ornithine. What you end up with is you consume 4 high-energy phosphates, ammonia, and bicarb. So this is what you're consuming, 4 ATPs, ammonia and bicarb, and you produce urea. And that's all that you produce. There's nothing else. Everything else is reutilized in the system. It operates, its only purpose is to get rid of excess nitrogen, okay? And that's all there is to it. And this is the end, the reaction. And here you have arginine, and you can see the makings of a urea molecule up here. Look at this, 3 nitrogens hanging around a single carbon center. That's a peculiar. Of course, the amino acid is in violet. You add water to that system, and out comes urea. And this is ornithine. And this is an essential amino acid. We cannot make that. So that also has to come out of the diet. And although you spend 3 ATPs to do this, you generate 2 moles of NADH, which is worth 6 ATPs. And so as you do this thing, not only are you creating the urea, but in the end, you actually generate energy. And this would be the full thing. Here's the arginine, here's the urea. And I want to point out that the nitrogen stuck on the ornithine doesn't move. That stays put. And you're going to resynthesize arginine and then cleave it again to get rid of the urea, and then remake it and cleave it again. And you just keep adding your nitrogens in through transaminases, typically aspartate in, fumarate out. Okay. Now, we've been there, okay? This is where we were. This slide is basically what we were talking about last week. So glutamate is the carbon skeleton of alpha-ketoglutarate with an ammonia group. The glucose can generate alpha-ketoglutarate off glycolysis in the TCA cycle. And then you can couple glutamate and alpha-ketoglutarate, and you can exchange them all you want. You want more, you want to get rid of the nitrogen, put it on the glutamate, it'll put it on the glutamine. You want to utilize, you need it for synthesis, take the nitrogen off the glutamate, stick it on the glutamine, move it wherever you want to use it. The glutamine is your storage form, pull it out of storage, put it in the enzyme reaction you want, consume the glutamine, and make that amino acid. And that's basically what's going on. And you want too much nitrogen, store it, okay? Need nitrogen, pull it out of the bank, okay? The bank is glutamine. The pathway into the glutamine is alpha-ketoglutarate glutamate. You just keep exchanging the nitrogens around. That's all you're doing is exchanging nitrogens. And just, again, generating NADH and trying to use as little energy as possible in one direction. And if you can, regenerate the energy in the other direction. And this just says the same thing, that basically that glutamate is the major source for all amino acid synthase. The thing is that glutamate can't cross the blood-brain barrier. We've been there before. We don't want glutamate coming in from our diet and changing our brain behavior. So then how does the glutamate get into the brain? Alpha-ketoglutarate through glucose. So all you need to do is you transport glucose into the brain. Glucose. Hello, you transport glucose into the brain. We did that. That was last week, right? But what do? You make alpha-ketoglutarate. Okay, then what? Transaminate it. Okay, just put a nitrogen on it and you have glutamate. What do? Do whatever you want with it. Okay, use it as a neurotransmitter, put it in the neurotransmitter pool, move it over to the synthesis pool. You don't like it, you want to store it, but you don't need it right now. Make it glutamine, stick it in an astrocyte, bank it. Astrocyte has more glutamine than it knows what to do with. Throw it out into the bloodstream, let it go to the kidney, pee out the pneumonia, and keep the carbon skeleton. What a simple and beautiful cycle. Okay, I mean it's an amazing cycle. Okay, but there are problems with it. Okay, we'll come to the problem. Here's glutamate at the center of the world. Okay, storage, enzymes, amino acids, neurotransmitters, carbon skeleton, energy, all running through glutamate. Okay. This is what we're talking about. Here's a nice picture introducing that. This is often known as the functional unit of the brain. So it's sometimes referred to the functional unit. It is the capillary, the astrocyte and its footplates, okay, and the astrocyte with its neuronal footplates. This is the business end. This is doing its business. This is the supply end. This is the converter, if you will. This runs all of the conversions so that the nervous system can remain pristine. We don't want neurons synthesizing glutamate, okay, very much. We certainly don't want them being involved in nitrogen, pathways, except as they need it, okay? We want them to be able to use it. The astrocytes surprise them with what? This is probably, there was a lot of people who would say, when we talk about the functional unit, this is the center of the brain. This is what makes the brain the brain. If you want to understand the brain from the intermediary metabolism viewpoint, this trio of blood flow, astrocyte, and neuron is the basic unit of the brain, okay? The neuron simply can't function without the astrocyte's metabolism, okay? And it is the astrocyte's job to make a function so the capillaries are tight, okay? But you don't, if it were totally tight and nothing could come or go, that wouldn't be very useful. So it's the astrocyte who controls what you take out of the blood and what you put into the blood, and all of the transformations that the busy, queen bee, if you will, is sitting there doing information transfer. That's what the neuron is about, is information transfer. You don't want it doing anything else that's complicated enough as it is. You want it to summate. You don't want it to have any other influence except the information that it receives from other parts of the brain. So to keep the information system pure, the astrocyte does everything, except what the neuron has to do for information transfer. That's the fundamental organization of the brain, okay? All right, and that's what I'm saying here in some detail. And by the way, I should have said this, I probably have, but I assume that you take an hour before next lecture, which unfortunately is tomorrow, but that you take an hour and review these slides. If you don't, I don't see how you could possibly understand everything. It'd be like my going through my Spanish lesson once, okay? It's not gonna stay, and not for me anyway. I have to go through it second or sometimes a third time. Simple. Glutamate to GABA is about we're one step away. You're always one step away from making all the GABA that you need, because you have all the glutamate you could possibly want, and it's only a single step. So that's GAD. Glutamic acid. Glutamic acid is glutamate. It's the same thing. The glutamic acid, decarboxylase. You just take off the carbon group, and that's it. Take off the carboxyl group, and instead of an amino, this is not an amino acid, is it? No, this is not. Look, this is an amino acid. This is not. GABA is not an amino acid. Okay, lost its carbonyl as CO2. And I like this diagram. It's nice and clean, returns us to synapse, shows us the important cycle. Here's the neurotransmitter cycle. I wrote an article for the Annals of Neurology in, I think it's second year, maybe 1982. And this was the topic, was this cycle, okay? Where you, either whether it's glutamate or GABA that's released into the intracellular space, extracellular space, if you're intracellular. Now here's GABA into the mitochondria, make glutamate, make glutamine. Of course, you have to get this back. You've got to get the glutamate back into the neuron so it can transfer it, but you don't want it doing anything more than it needs. You can't transfer the glutamate. You don't want your neurons. You want your neurons to generate and control their own glutamate levels. Doesn't even want to be dependent on the astrocyte. So you move up one step. But anyway, glutamine is a great transfer molecule because it's neutral, it's not ionized, it just travels so easy, it has nice dielectric constant, it just travels easily via transporters. You put the glutamine in, if the neuron wants to make glutamate now, it makes glutamate. If it doesn't want to make glutamate, it doesn't make glutamate. So it controls its own fate. And I put this picture up, this was from a few weeks ago, because I want to re-emphasize the role of mitochondria. I think I had emphasized the importance of energy metabolism in synaptic function. But now I want to return to it and say the mitochondria wasn't only there because it can generate a lot of ATP. The mitochondria was there because the nitrogen metabolism for the synthesis of these neurotransmitters is highly dependent on the mitochondria. So the synthesis of the reloading of the making of glutamate and GABA all depends on the mitochondria as well. So they're not just there for ATP, okay? They're there for a couple of reasons. And then this diagram looks confusing. It's really not. It just shows you that you can have, here's the glial cell, here's a glutamatergic neuron, here's a GABAergic neuron, here they're doing their thing, here they're doing their thing. They both come into the system. The GABA has to run into the mitochondria to come back out as glutamate, actually runs through the citric acid cycle, comes out, alpha-ketoglutarate, of course, it doesn't come out as glutamate. It comes out as alpha-ketoglutarate. Why do we write glutamate? Because who cares? You just transaminate it and it's glutamate. So nice and easy, always there, okay? Another picture of it, sharing the fact that that there's this parallel neuron to glia to neuron. So the neurotransmitter goes from neuro to glia. The glia uses glutamine. And we're almost done. I just want to end up now with a discussion of hepatic encephalopathy, which is really important. And this is one lecture where it makes no sense not to talk about clinical, because the clinical tells you a lot about the basic as well. So there's a strong interaction here. We get into trouble with hepatic encephalopathy. Obviously, the liver is failing, or the kidney is failing. The urea is not being excreted. Okay, that's the basic problem, is we're not excreting urea. In one case, if the kidney is failing, then the liver has been making urea, but it accumulates. The UN goes up, urea and nitrogen. In the other case, it doesn't go up, right? In a hepatic failure, urea is not made. Then you have ammonia and ammonium floating around in the bloodstream, right? You could say, well, why doesn't it use the glutaminase, glutamine? The liver's failing. So not only this isn't one of those acidotic situations where, okay, I'm not going to use my bicarb, I'm going to the liver turns to glutamine. No. The glutamine is the liver's shot. So what other organs are there that can deal with nitrogen in the body? Well, in order to have your own nitrogen metabolism, you basically have to be sequestered from the rest of the amino acids. You have to have a reason to do that. There's only one organ that does that, and that's the brain. Everybody else just depends on the liver for this balance of nitrogen. Okay? It's only the brain is the only alternative that the body can turn to. And so you find your brain, in times of hepatic failure, the brain becomes the major organ for excretion of ammonia. That's not what it was designed for. Okay, but that's what it's going to do. Because the high ammonia and ammonium drive the system by mass action of your enzymes. It drives the whole brain toward glutamine synthesis, because that's how you excrete glutamine, nitrogen. And at times of excess, and the whole brain is experiencing the excess ammonium because it's everywhere. Okay, this is not normal anymore. It's got an excess of nitrogen. That pushes all of the enzymes in the brain to synthesize glutamine, which then is released by the brain into the bloodstream and hits the kidney. If the kidney is working, at least the kidney goes, fine, I don't know why I'm seeing all this glutamine, but out it goes, and it gets rid of the nitrogen, goes back to alpha-ketoglutarate. But here, Michael Rittenuti, one of the residents at this lecture 2 or 3 cycles ago, Michael asked a question, and he said, excuse me, but that's all well and good that the glutamate is regenerated by the kidney. But glutamate can't cross the blood-brain barrier. And we thought about it in this room, and we said, right, it will have to create new glutamate. So it's losing, when it excretes nitrogen, it excretes glutamine. When it excretes glutamine, it excretes glutamate. If it's doing that for the entire body, it's going to run through a lot of glutamate, which means you're going to have to resynthesize glutamate. Okay, you say, well, glucose, alpha-ketoglutarate, make new glutamate. Hello, okay? We're now in competition with energy metabolism. So the glucose is being diverted from the brain for glutamate synthesis in order to handle the nitrogen load of the body, of the entire body, okay? Therefore, nitrogen metabolism is in competition with energy generation in the brain. And it stands to reason, okay? This has never been proven. We have tried to design experiments with the liver team here several times. And eventually we will. but it stands to reason that the brain's glucose utilization is going to go up during hepatic failure, not because it needs more energy, but because it's consuming the glucose to run the ammonia cycle, which causes it to excrete glutamate out to the kidney through glutamine, but can't take it back up. And so the glucose that's used has to both run the brain's metabolism, which is fast, and excrete all the nitrogen for the body. And that's a challenge. And it's not going to do that if the serum glucose is 90, right? So basically that's it. Let me just finish with this. The older theories of the pathophysiology is that it was an osmolar load from glutamine. That makes no sense at all. First of all, under any circumstance, you have only 1 to 2% of the osmoles with glutamine, and that idea is wrong. And I don't know what it's going to be replaced with. People are thinking that there might be from free radical. I'm telling you, I believe that it is a glucose failure is actually what the problem is. All right, that's it. Yeah. We're done.
Simon and Stephen are in same place.
Mhm.
They are logged in.
Okay.
Listening to the lecture. So I was just wondering if we could keep doing that when people are at the same place, since they still have access to
Can
the
they
lecture.
see the slides and hear at the same time?
Absolutely.
Okay.
And so that's what they
Yes.
are actually doing right now.
I'll say yes to that.
Okay. Because that
I don't love it. I got to tell you, I'm not in love with it. I don't think it's quite the same, but close. It's close enough.
Okay, so we can keep
Yeah.
doing that. They're listening right now, so that's
Hello.
I wanted to make sure that they were.
Yeah.
So tomorrow they'll log in again remotely.
Right.
And
Now today, yes. Today we end the two lecture series on intermediary metabolism, and the next section that we get into is on signaling. So we are now going for the rest of the year.
Steven says hello.
Hello. For the rest of the year, we are going to be talking about the mechanisms by which neurotransmitters act. And then next year, meaning January next year, we will, for the rest of that year, we will look at the neurotransmitters in various forms, okay? But in the next 5 lectures, I think, we're going to talk about the underlying signal cascades that are available to neurotransmitter receptors, okay? G proteins, phospholipases, phosphokinases, the acyclic nucleotides. Those will be the topics that we'll be covering in the next 5 or 6 lectures. Okay? That's the way the course is organized. It's actually, I got to tell you something. I wish somebody taught me this course. This is in my opinion, this is one heck of a neurochemistry course. It's really pretty slick. I gotta say, I just think it's a serious course, and it must feel like a serious course. But I think that it's important that we put this mini symposium together so that we take it out of the abstract and make you use it. And I think when you use it, you will see how much you've learned. I don't think you realize how much you've learned in this course. And I was saying, like, I think on our website, on the residency website, we should put some URLs to our YouTubes. Pick out the 2 or 3 best YouTubes. They could be on any topic you want. I don't really care. Or take a smattering of topics. Take one of these basic lectures, a couple of good clinical lectures that you've done. Yeah, statistical, whatever you feel like. Put a smattering of this stuff out there. Just put the URLs on the web, so on our website, and then we'll look very high tech, by the way. We'll look so cool. Okay, and it will also jazz up the website, which looks like a piece of poop to me.
Yeah, here. So it's done. Okay, we're good. Cool. We're done.
Thank you.